Hello wonderful people, another day, another video. Today video is going to talk about acute and chronic inflammation. Usually inflammation is the body defense mechanism in which the pathogen alive in the body, and then they use this innate immunity to fight against those pathogens. Usually inflammation is body defense response to injury or infection marked by redness or swelling or heat, pain and tissue repair. So means those are characteristics of this inflammation. Inflammation are of two types, acute and chronic inflammation. We're going to say acute inflammation when there is the duration is short time, but if there is a persistence of the infection. So without delaying, we should start by knowing that acute inflammation usually is good because our target is to come and eliminate the pathogen which invade our body. So if the pathogen become persistent, it's going to be bad because we are going to have the chronic injury or chronic damage of the tissue. So let we have the detail on the acute and chronic inflammation. So actually there is the classification where acute and the chronic inflammation, there are difference. We will talk about acute inflammation when you have rapid onset, to mean that there is the way they're going to be okay, they're going to be rapid, while the chronic, it is later onset. It means the symptoms, they are going to come later. There is what we call short duration. Chronic inflammation, it is the longer duration. Hallmarker of the acute inflammation, it is edema. We will see what edema is. Now the hallmarker of the chronic inflammation, it is granuloma formation. And then the immune cells which intervene in the acute inflammation, it is the neutrophils, while the immune cells which occur in this chronic inflammation is called macrophage and the lymphocytes. So this classification, it is very important because when they ask you to differentiate acute from the chronic inflammation, we refer to this table where rapid onset, short duration, edema, neutrophils, that these are in the acute inflammation. While later onset, longer duration, granuloma formation, and macrophage and lymphocytes, all of these are chronic inflammation. So, without delaying, let me talk about now acute inflammation. So, let me have the marker. So, means our target now, we're going to talk about acute inflammation in detail. So acute inflammation usually it means that antigen, it is entering in the body. After entering in the body, we're going to create a way in which the neutrophils, as we have seen that neutrophil is the one which mediates this acute inflammation. We're going to see if how the neutrophil can come from the blood vessel toward where there is the problem or there is injury. So what we are seeing here, let we talk about on this diagram. This is usual blood vessels. This is blood vessel as usual. Means after antigen arrival, this is the antigen which is arriving. It releases what we call mediator release, or for example, histamine, which is released by master cell of the infected area. Now this master cell, they are going to be released in the blood flow. So the first thing of this acute inflammation, we are having 5 separate events, which is very, very important to understand. First things, it is what we call transient vasoconstriction. Means the blood vessels, they're going to be transient vasoconstriction. Means they're going to constrict, but it is a short time. The next, they are going to have what we call vasodilation. So this blood vessel, after vasoconstriction, automatically we're going to have what we call persistence vasodilation. Persistence vasodilation. This is persistence vasodilation. So means after this persistence vasodilation, we are going to have what we call increase of hydrostatic pressure. To means that this blood vessel, after enlargement, they're going to have accumulation of the blood. It means many blood flow here, blood are going to be accumulated here. Now after being accumulated here, they are going to be what you call increase of hydrostatic pressure. Remember, in the blood, there is a lot of water. Means if there is a lot of water, means hydrostatic pressure is first due to the high amount of water. Now they're going to be start by leaking out. Fluid, we're going to start leaking out. Fluid, we're going to start to leaking out. Now, after increase of hydrostatic pressure, also there is what we call increase of vascular permeability. So, in the vascular, there is what we call the endothelium cells. Between one cells and another cells, there is a pore. Now, after swelling like this, we are going to have what we call, this pore will increase. Now, this will increase what we call vascular permeability, so that the white blood cells can get the gate to enter in this damaged tissue. So, on this diagram, you see, usually this is the blood vessel. Now, after blood vessel, they are going to be vasoconstriction, but short time. Vasodilation is going to be persistent like this. This is vasodilation, swelling. So here there is swelling, where in the characteristics of acute inflammation, we have what you call swelling. Here also the redness, many more blood in this area will cause this tissue to become red and also to increase the heat, where we have high fever in the inflammation. So as you see, this is the endothelium cells, okay? Means after increase of hydrostatic pressure, which will make the fluid to be out, and also after time, they're going to have what you call, this is what I have said, the increase of vascular permeability. Now here there is a stasis of the blood. Now the neutrophils are going to intervene to pass in those pores. So this is the overall the story of this acute inflammation. So let you have to look on the theory. Means there is step number 1 is the antigen arrival. Step number 2, there is what we call transient vasoconstriction. This is number 2. Means the blood vessel, they're going to do constriction. After it is transient because it is short time. The next step, they're going to be persistence vasodilation. Means persistence vasodilation, they're going to vasodilate like this. Means after this, they are going to be accumulation of blood in this area. So accumulation of blood in this area will cause this tissue to become red in color, because blood is red, and also increase heat. Those are the two characteristics of acute inflammation. So next, after this vasodilation, in the blood there is water. There are going to be increase of hydrostatic pressure. Now hydrostatic pressure will cause edema. Also, it is another characteristics of acute inflammation, where they're going to call transudate. Means the fluid will leak out this blood vessel, but this fluid, they contain only water. There is no proteins. So it means, protein are large. Because protein are large, can't penetrate this endothelium pores once there is no increase of the vascular permeability. So after, means increase of hydrostatic pressure will cause the water to leak out this blood vessel, which will cause edema, but this edema, it is transudate edema, there is no proteins. So the increase of vascular permeability means they're going to have like this, means the pores are going to increase. Now the protein can go out of this blood vessel, which we call exudate edema. So exudate edema, it is the body fluid are going to leak out the blood vessel, but they contain the proteins and other large molecules because of increase of vascular permeability. Now after this, they're going to be what we call blood stasis, where the neutrophils now are going to intervene. So those are 5 steps of acute inflammation. Number 1, transient vasoconstriction, persistence vasodilation, increase of hydrostatic pressure, increase of vascular permeability, and stasis. So let we now go on the events of the cellular events. So in the acute inflammation, you have the cellular events, but the starting points, it is the vascular events. Now, in these cellular events, we are having all 6 steps, so that we can engulf bacteria which coming now to end our acute inflammation. Those are the 6 steps of the acute inflammation. So, step number 1, it is what we call margination. But let you have a look on the figure. So this is blood vessels, as you are seeing here. This is blood vessels. So in these blood vessels, usually blood are flowing as usual. There is what we call axial blood flow. So axial blood flow it is where the blood, it is like this, in this way. Now the heavy blood cells are in the middle, where means white blood cells are in the middle. Now red blood cells are flowing. Red blood cells are flowing. What is flowed? It is platelets. Platelets. Now the plasma, which is lightest one is now on the peripheral one. So this is what you call axial circulation. Now our target is to take these neutrophils toward where there is a pathogen. So this happens in 6 steps. Step number 1, it is margination. Margination is to change axial blood flow to reverse axial blood flow, where red blood cells can now flow on the peripheral. Now this is means margination. It is turning the moving of red blood cells from the center of the blood vessel to the peripheral, where now this process is called margination. So after margination, we are going, means this is where the blood cells, the gate, it is here. So it means that after margination, you should take this white blood cells now transporting to this gate, where it can interlink to the antigenic point. Now the process of moving these red blood cells in this way are called rolling. So after rolling, now the cells, they reach on the gate. After reaching the gate, they will be what you call adhesion. All of this process are making them possible because of receptors. Some receptors here on the wall of blood vessels, and some receptor of these neutrophils, and so on and so on. So this adhesion means the neutrophils, they reach on the gate where it will going to attack the antigens. So now the process in which this white blood cells are going to be transported from the blood vessel toward the tissue is called diapedesis or transmigration. Means it is a cross of white blood cells across the endothelium wall. So means macrophage, it's now reached here. So macrophage do not have eyes. There is no eyes on macrophage, or there is no eyes on the neutrophils. So how the neutrophil will know that the antigen is in this direction? So it will help by what you call chemotoxins. So chemotoxins movement, it is the unidirectional movement where these neutrophils or macrophage, they are going to throw the certain chemicals like C5H. It is one of the complement which act as in the chemotaxis, which will go and then bind the antigen, which is here. Now after reaching there, by help of chemotaxis chemicals, they're going to do what you call phagocytosis. They will engulf and then they will eat this bacteria. Now acute inflammation is ended, because our target was to change this vascular so that we can create the gate of this white blood cells. Step number 1, it is the acute, it is transient vasoconstriction. The next, it is persistence vasodilation. The step which is the next, they are going to be increase of hydrostatic pressure, increase of vascular permeability. Now we are having what we call stasis of the blood. In the stasis of the blood, in increased vascular permeability, the gate is now created. Now, let we talk about the vesicular now in here. Now in the vesicular, step number 1, it is margination, change of axial blood flow to the peripheral. Neutrophils now in the peripheral, they roll itself toward the gates, which is where the epidesis occur. Now by chemotaxis, now those phagocytes will engulf the pathogen. Now pathogen is killed. So that's all about acute inflammation. So let we talk about the chemotaxis movement, where we are having like a chemicals like leukotriene B. Here this is chemicals, it is help in this chemotaxis movement, where neutrophils will detect exactly where there is pathogen. As I have said, this is the complement which is helping there. And also there is some cytokines which help there, like interleukins, and particularly what you call interleukin 8. All of those molecules, they are helping us in the chemotaxis. So this one, it is complement 5A. It is most powerful chemo attractive or chemokines which orient phagocyte where there is a pathogen. So I hope in the Acute inflammation, you are aware of all of the vascular events and also cellular events to fight against this pathogen. So what will happen if the antigen now is going to be persistence? If there is persistence, we have seen that in the chronic inflammation there is later onsets. We will say form what we call granuloma. Now monocyte and macrophage and lymphocytes are the cells which intervene there. So now let me see how the steps are flowed. So usually they are going to be a rise of the antigen. So if the antigen is a rise, it will meet with antigen presenting cells. So these antigen presenting cells will present this antigen on helper Th1 cell. It is not helper T2, it is helper T1 cells. This helper T1 cell will recognize that this antigen is non-biodegradable, or this antigen it is a persistence. For instance, like TB, Mycobacterium tuberculosis, also it is responsible for this chronic inflammation. Chronic inflammation also is the same as granuloma inflammation. So here, what will happen? This helper T2 cells, it will secrete what we call interferon gamma. This interferon gamma will recruit monocytes from the bloodstream. These monocytes, when they reach the tissue, it will become macrophage. This macrophage, there will be serial changes where macrophage will become epiteloid cells into giant cells. Not only this one, which is secreted by helper T1 cells, but also it will secrete what we call interleukins 2, which will recruit immune cells, another immune cells, which is, for example, another helper T1 cells. What is the function of those immune cells is to come and make wars on this antigen, so that even if this antigen is not be able to be destroyed, but this antigen cannot harm our body because it is formed the war on itself. So let me have a look on the steps. Steps of the chronic inflammation. Number one, it is persistence antigen stimulus. As I have said, there is T helper cells which is going to be activated now by help of antigen presenting cells. It will release interferon gamma, interleukin 2, and tumor necrofactor alpha, which produce those cytokines which is released by this helper 1 cells. Means this one, it will recruit macrophage. Macrophage will be activated and macrophage will change to epiteloid cells formation. After forming epiteloid cells, some macrophage will come to form what we call giant cell formation, another form of the macrophage. Not only this, but also this interleukin 2 also will come and now recruit another immune cells which is called T cell infiltration. Now there is a tumor necrotic factor also will recruit what we call fibroblast, which is going to help to make encapsulation by help of the collagen, which is formed by this fibroblast. And now pathogen, now it is now protected by those immune cells. Means antigen is like this. Now the macrophage, like which is giant macrophage, and even epiteloid cells will come and now surround the pathogen. Not only this, also T cells will come and bind on this pathogen, which is undegradable by the easy way, and also they're going to form what we call fibroblast, which come and form the collagen, and now this collagen is going to form like a capsule, which form like envelope of this immune cells together with the antigen. Now this is what we call granuloma formation. So this is what all about the chronic inflammation. Chronic inflammation also, their end product is granuloma formation. Means that once you understand the chronic inflammation, you are able to explain granuloma formation. So I'm going to talk a little bit on the granuloma formation in the summary. Point number one, it is persistence irritant or pathogen which trigger chronic inflammation. This is number one. Antigen which come here, as I have said, it is the going to be persistence. Okay. After this, macrophage will fail to clear the irritant. Means macrophage will fail to clear or to phagocytes this antigen because it is non-biodegradable. Now they will activate T cells. Which T cells? Helper T1 cells. This is another step. After this, we are going to have a T cell will release interferon gamma, which will activate the macrophage. And we have seen that this macrophage is going to change the epithelioid cells and the giant cells. This macrophage will be transformed into epithelioid cells or giant cells. What is the next? There is what called fibroblast, which deposits collagen around the structure. Now granuloma is going to be formed. Necrosis may be occur in the center, and then this type of granuloma called CSS granuloma. This is an example for TB, Mycobacterium tuberculosis. They form what we call CSS granuloma. Now, this is what's all about the summary of granuloma formation. The same case, chronic inflammation. So I cannot end without talking about the granuloma structure. In the structure, as we have understood that there is antigen in the structure. This is the antigen. Now it is surrounded by giant cells and epithelioid cells. Some epithelioid cells. There is some T cells. T cells are float. T cells are float. Now after T cells, we have what we call collagen protein, which formed by fibroblast, which come and make encapsulation of this pathogen. So in the core, pathogen with or without necrosis, this is what is happening in the core. Now epithelioid cells transform the macrophage will now flow. Giant cells, which is fused the macrophage, also will be flowed, as I have talked about. Now T cells or lymphocytes will come and now surround the antigen if the necrosis is there, epithelioid cells or giant cells, and even T cells or lymphocytes. Now fibroblast capsule, which come and form collagen, and fibroblast will enclose this granuloma. Now we have the word diagram of granuloma. If they ask you to draw how granuloma formed, even you can explain this and you are done. So thank you for your kind attention. This video, I hope it is very helpful. If it is helpful, you can do subscribe, comment and share, and also thank you for watching.